Hello, my name is Brandon Wood. I'm an Associate Director of Regulatory Affairs at Teva Pharmaceuticals. I have been a regulatory professional for over 10 years, and I specialize in complex generic products, including peptides, iron colloids, long-acting injectables, and drug-device combination products. The title of my presentation today is Implementing GDUFA Science in Product Development and ANDAs, Realizations and Recommendations. This is our standard disclaimer slide. In terms of presentation contents, we'll start with an industry perspective on the GDUFA Science and Research Program. Then we'll discuss realizations and recommendations for complex mixtures and peptides, complex injectables, formulations and nanomaterials, drug-device combination products, and long-acting injectables. And then we'll go ahead and wrap up with some parting thoughts. I'd like to start with an industry perspective on the GDUFA Science and Research Program. In summary, the Generic Drug User Fee Amendment, or GDUFA, established a science and research program at FDA that is implemented through extensive intramural and extramural research collaborations. The program supports the development of innovative methodologies and more efficient tools to help establish drug equivalence standards and support the development of safe, effective, and high-quality generic products for the American public. This research is particularly important for certain pharmaceutical products known as complex products, which are harder to develop as generics. Complex products often have fewer generics or none at all. In the absence of market competition among generic alternatives, these medicines can be so expensive that patients who need them may not be able to afford them. From an industry perspective, it's clear that the realizations of this program, such as product-specific guidances, general guidances, scientific articles, posters, characterization techniques, bioequivalence approaches or considerations, etc., are vital to successful development programs for complex generic products. There is certainly alignment on the topics and initiatives that can greatly benefit from additional research. And also, this program will be fundamental in addressing technical uncertainties and challenges that continue to arise for these complex product types, thus enabling more timely approvals. So now we'll move on to realizations and recommendations, and our first product type here is complex mixtures and peptides. To start with an overview, in fiscal year 2021, research efforts continued and the development of advanced analytical methods for the evaluation and characterization of complex active pharmaceutical ingredients, including complex mixtures, oligonucleotides, peptides, and synthetic polymers. Within Teva, characterization of complex APIs using advanced analytical methods is essential for supporting a demonstration of pharmaceutical equivalence and linking product attributes to safety, quality, and clinical performance, thereby facilitating the generic drug development and approval process. Moving on to realizations. In fiscal year 2021, FDA issued the final guidance, ANDAs, for certain highly purified synthetic peptide drug products that refer to listed drugs of rDNA origin in May 2021, issued 2 new PSGs related to complex mixtures and peptide products, published 1 new article, and facilitated 2 posters as well as 6 presentations. I would like to take a second and make 2 notes. All overviews and realizations in this presentation were taken from the fiscal year 2021 GDUFA Science and Research Annual Report. And I would also like to note that the realization here and presented throughout this presentation is in addition to the new, continued, and completed grants and contracts, as well as active FDA research. So while acknowledging the great realizations for these product types, I'd also like to explain some challenges, examples, and provide a recommendation that may be useful. Challenge with respect to complex mixtures and peptides is that new impurities or impurities that are also present in the reference product, but at a higher concentration, are handled on a case-by-case basis using a totality of evidence approach, including in vitro immunogenicity studies. In vitro immunogenicity studies are becoming increasingly prevalent for these product types. However, there is limited guidance available for study design considerations and expectations. Just to provide an example, only after multiple review cycles for one product did we learn of a minimum dilution level for samples to be tested in innate immune assays. This new information was not previously discussed in guidance or pre-ANDA feedback, and this previously unknown requirement impacted both completed and planned studies. As a recommendation, from a public domain standpoint, if FDA could develop multiple publicly available validated methods for innate immune assays and DCT cell assays that the agency finds acceptable, this would likely reduce the number of deficiencies in CRLs. Additional specificity and guidance for study design considerations would also likely reduce the number of review cycles. So moving on for realizations and recommendations, our next product type is for complex injectables, formulations, and nanomaterials. In fiscal year 2021, the agency's research efforts for complex injectables, formulations, and nanomaterials focused on the following aspects. 1. Development of novel in vitro drug release test methods. 2. Evaluation of analytical methods for characterizing complex injectables. 3. Use of in silico multi-scale model to evaluate target site bioequivalence and critical quality attributes of injectable nanomaterials. And 4. Investigate the relationships between physicochemical features of amphotericin B liposomes and product toxicity. From a Teva utilization perspective, characterization of complex injectables is required to demonstrate in vitro bioequivalence as well as drug substance and drug product sameness. Teva reviews all PSGs, articles, posters, and presentations issued by the agency to ensure that there is a common understanding of the techniques and expectations for advanced characterization tests. In fiscal year 2021, FDA issued 4 new and 2 revised PSGs, published 4 articles, and facilitated 7 posters as well as 11 presentations for these product types. Moving on to challenges, an example, and a recommendation for complex injectables, formulations, and nanomaterials. As a challenge, I would like to note that there are no publicly available analytical methods for in vitro characterization established by FDA. What we see is deficiencies in CRLs often relate to the way products are characterized, and insufficiency of characterization. Only during the review procedure is FDA requesting industry to follow very specific methodologies. We'd also like to note that the requirements evidenced in various PSGs are continually changing, and the in vitro parameters that impact in vivo performance are debatable. To provide an example, for one product, Moss power spectroscopy data was provided for physicochemical characterization. The samples were lyophilized. Only after multiple review cycles did the agency request the analysis be done on frozen samples instead of lyophilized samples. This specific sample preparation was not previously discussed in guidance or FDA feedback and could have been avoided if the preference was known. So as a recommendation, we'd recommend that the agency conduct research to publish publicly available analytical methods with sufficient level of details for in vitro characterization and/or develop guidance to communicate FDA's preferences in study designs specific to complex injectables, formulations, and nanomaterials. And we'd also recommend that the agency facilitate further evaluations of in vitro parameters that are clinically meaningful. Next, for realizations and recommendations, we'll move on to drug-device combination products. In fiscal year 2021, FDA continued to perform research related to the impact of identified user interface differences on the therapeutic equivalence of complex generic drug-device combination products, or DDCPs, and their reference listed drugs. In the absence of a final guidance, Teva has utilized numerous pre-ANDA meetings to work with the agency to develop acceptable comparative use human factors study designs to justify other differences present in the final user interface. For realizations in fiscal year 2021, FDA issued 1 new PSG that was directly impacted by GDUFA funded research in this area and facilitated 9 presentations related to drug-device combination products. Moving on to challenges, example, and recommendation. One significant challenge for drug-device combination products is that there is no available guidance that represent FDA's current thinking on comparative use human factor studies, or how to properly calculate a non-inferiority margin to employ in a comparative use human factor study. I'd like to note that there is a discussion of non-inferiority margins in the draft guidance for industry comparative analyses and related comparative use human factor studies for a drug-device combination product submitted in an ANDA dated January 2017. However, as per recent feedback, this draft guidance does not represent FDA's current thinking. For one drug-device combination product, as an example, we submitted a controlled correspondence requesting feedback on our comparative use human factor study protocol. FDA responded, strongly recommending a pre-ANDA meeting, and we took the advice and filed a pre-ANDA meeting request. Later, the meeting was held. However, it was recommended to revise the protocol and submit another controlled correspondence. In total, it took 14 months to get meaningful feedback, and I think the agency would agree that that is not a conducive timeline for generic product development. So as a recommendation, regulatory science and research regarding acceptable study designs and non-inferiority margins that can be employed in a comparative use human factor study, including workshops, training, and focus groups, would be beneficial to FDA and industry. Research in this area could facilitate a common ground whereby other differences between test and RLD combination products could be effectively managed and not preclude approvability via the 505J pathway. Now, I would like to note that even within this public workshop, there is another session on comparative use human factor studies. Our final realizations and recommendations are related to long-acting injectable products. As an overview, in fiscal year 2021, research projects on long-acting injectable and implant products aim to 1. Develop new tools for characterizing complex polymeric excipients, 2. Better understand the impact of variation in raw materials on formulation, physicochemical characteristics, and drug release, 3. Explore new in vitro drug release testing methods that have better clinical relevance, 4. Investigate advanced imaging tools to explore correlations between formulation characteristics and in vitro in vivo drug release, and 5. Develop new modeling tools to support the development of alternative bioequivalence approaches. Teva has utilized numerous pre-ANDA meetings to work with the agency to develop acceptable modeling and analysis plans as alternative bioequivalence approaches for long-acting injectable products. For realizations in fiscal year 2021, FDA issued 4 new PSGs, published 15 articles, and facilitated 12 posters as well as 5 presentations for long-acting injectable products. So to describe some challenges along with an example and a recommendation for long-acting injectable products, I would like to note that FDA has indicated an interest in seeing modeling and analysis plans in terms of model-based approaches for bioequivalence assessment, if proposed, as part of pre-ANDA submissions to obtain concurrence prior to execution. It has been highlighted that information requests are common for these types of pre-ANDA meetings and take time away from the assessment clock. Model integrated evidence can have a meaningful impact on reducing study durations, but the specific expectations are unknown. Teva has now submitted multiple modeling and analysis plans for the agency's review. Without an understanding of the specific elements that FDA would like to review for modeling and analysis plans, these pre-ANDA meeting packages often have 75+ attachments, meaning we provide everything from scripts to individual datasets. Really, it's unknown if this full package is useful for the agency's review or we are providing too much. So as a recommendation, it would be beneficial to both industry and FDA if there was a mutual understanding of the information to be submitted in such pre-ANDA meetings to make the most of the meetings and set a strong foundation for the alternative bioequivalence data to be generated in support of an ANDA. Additionally, we'd recommend to develop guidance specific to model integrated evidence with the suggested approaches, designs, and templates for submitting the information in a pre-ANDA meeting package or an ANDA. To wrap up with some parting thoughts, the agency's output from the GDUFA Science and Research Program will inevitably enable a stronger environment for complex generic product development. Continued feedback and industry-agency collaboration will streamline the utility of the research performed, and focus should be put on ensuring that the research performed is compatible, useful, and reproducible for generic applicants to incorporate into development programs. Detail is key. And now to close with a quote by Winston Churchill that I thought was very appropriate. Out of intense complexities, intense simplicities emerge. Thank you.